heard you were overjoyed that you thought I was dead.
Oh no,
I was hurt when I saw that text. I cried to sleep and then woke up and decided to pull the fuse off me.
Bro didn't care that I had died.
But it smells just like
What's
Not
better,
at all.
being remembered after death, but it's in like a pitiful way, or just not being remembered at all?
Well, I mean, if you're being remembered, you're not.
Hey, look,
Not being
if you're remembered, that means you're a legend. In the wise words of someone from the Sandlot. No, wait. No, it means you're a hero.
All right, let's see. Is it time to get started?
Legend.
It is time. All right, welcome to class, everybody.
Yeah.
Woo!
Yippee.
Pi Day, everyone. Yep,
Yeah, I didn't get any
Pi
fires
Day.
going.
Happy DNA extraction from food, meiosis, and genetics day. It's a good day. Lots going on today.
No, that was my least favorite.
Becca, do you have a question?
Aaron.
Do you have a question?
Aaron. I thought you were
No,
gonna
no.
She was just making movements.
Yeah?
She was excited.
Yeah.
She was excited.
You know what I mean?
All right,
I do-
Deanna's going to make an announcement about something cool happening.
Whoa. Yo, everybody. So, I don't know, you probably know this, but I work for the Shasta Birding Society and I do their social media. And it's my job to get you young people out there and looking at birds. So, this Saturday in Red Bluff, we are doing a walk from 7:15 to noon. Now, that sounds really early, but I think that we're actually starting at like 8. So, 7:15 is for like transportation, right? It's a 3.5-mile hike at the Iron Canyon Trail, and we're gonna see a bunch of really dope birds. There will be binoculars if you don't have a pair. If you'd like to borrow a pair. I think Becca might be there, maybe with me. So yeah, come out. It's free. Birds. Learn about birds in your environment. It's very cool. Thank you.
I didn't know you were a tree.
Yeah, do you know the greenhouse key though?
Like this is a nice water bottle, even though your name is Josh, because Joshua Tree. But like Garrett's water bottle is lame.
What's Garrett's water
It just says Garrett.
What did I got after you left?
It looks like an AIM.
Yeah,
Oh.
smell like pepper.
Baby food.
What dinosaur?
And you have an added green for each dinosaur.
I saw a completely random like crossover and I forgot what it was.
Fallout?
Yeah.
Yeah.
I'm like,
Double decks.
Yeah.
I just didn't get it.
That just
What
happened.
does Fallout have to do with
Nothing.
crossover?
No, they just
I
did
just
a
think it's
crossover.
fun.
One of their units versus Beyonds. They're doing Marvel next year.
They're doing, yeah, they also just did Doctor Who.
Yep. And they've done Lord of the Rings. They've done
Lord of the Rings makes
They've done Jurassic Park. They've done Transformers. They've done Fortnite. They've done Princess Fortnite. They've done Street Fighter.
Are you talking about like? Wait, what is he talking about?
Magic the Gathering crossovers.
Oh, it's like Yo Transformers.
Or any other parts like good.
Some of them are actually pretty good. One of the Transformers ones is actually used.
Bro, getting hit by a transformer in a competition is so bad.
I mean, I haven't heard that.
Did you check out the notes I sent you?
I did not read them.
Did you check out the video of the Air Up bottle I recommended?
I actually watched it before that.
Oh, you didn't say anything.
I did.
What did you think about it? I thought it was pretty sick. That's all.
It is, yeah.
Like it wasn't like an extraordinary video. I just didn't realize they were that bad, and I just thought that was goofy.
Imagine making
Me. First, like obsessing over every seed.
I don't write with my left hand.
You always write on your left side though. You'll do this.
Yeah, I literally.
Why? You might as well be left-handed.
It's on both sides. I do it with both.
No, every time though, you're like normally over there. The only reason I feel like it's
Work here. If there's something in front of me,
yeah.
What is the?
I'm working over here. If I'm
I mean,
like looking anywhere else, and that's just what's comfortable.
See how we do time-wise. We've got next week to cover genetic modification, continue to talk about DNA and genetics as needed. But ultimately, the plan today is to discuss meiosis, alternation, regeneration. And genetics, we're going to be introducing genetics and introducing DNA, RNA, which these topics will also roll into next week, okay? So we want to make sure we have a really good grasp on this topic, which shouldn't be too hard for us, I don't think, because we already covered mitosis, and meiosis is like mitosis on steroids. It's just a big extension of what's going on with mitosis, and it's super important to understand in any biological science kinds of classes. You'll probably cover this topic at some point. Remember what we talked about keeping the chatter down so that everybody else can pay attention to. Alteration of generations. We already had an introduction to this topic when we had the Bryophyte Day. And alternation of generations is, general life cycle of plants, because plants do this really crazy thing that there's like an extra, like whole level that happens in the life cycle of plants, and that animals don't have. It's as if we think about our sperm and egg developing into multicellular organisms, and either still connected to us or disconnecting from us and living on their own, and then they develop the egg and sperm. That's weird to think about it that way. But it's plants do something like that, which is crazy. We'll get into that. And then once we cover every single major plant group that we cover in our next big section, which is diversity of plants the part of the semester after we finish plant physiology, each plant group, the bryophytes, the pteridophytes, lycophytes, and the gymnosperms and the angiosperms, we're going to be hitting on the same thing, alteration of generations. So if you don't fully get a grasp of it at this point, I want you to get the general idea, and then we'll get into it in detail of like how it's different between each of those major plant groups. And then of course genetics is DNA, RNA. So we have a little activity to do for practicing meiosis, and then we the end of class today will be getting a chance to extract DNA from food items to get ready for continuing that process of trying to figure out if there is any plant DNA in that food, first of all, and then also whether or not there's any of those markers that we're going to be looking for that can signal to us that the plants used in that food were genetically modified. So, and that's gonna extend into next week. All right, so to introduce things first, let's watch this short video on an overview of meiosis to get an overview of it, and then we'll get into it in more detail. Make sure I got my volume up. That's you.
In this lesson, we'll look at an overview of the type of cell division called meiosis. Meiosis takes place in an organism's reproductive structures, called gonads, for the sole purpose of producing haploid gametes that are genetically different. These haploid gametes are sperm cells in the male and eggs in the female. In the plant kingdom, pollen grains contain the male gamete, while structures called ovules contain the female gamete. Let's take a brief look at how meiosis produces gametes. A cell about to undergo meiosis will have already replicated its chromosomes during interphase of the cell cycle. This original cell is diploid, which means it has 2 sets of chromosomes, one from each parent, sometimes written as 2N. Through 2 stages of cell division, meiosis produces 4 genetically different haploid gametes, sometimes written as N. For this reason, meiosis is also called reduction division. It reduces the total chromosome number in half. So, when the haploid sperm cell and haploid egg cell unite to form a zygote during fertilization, the diploid number of chromosomes is restored in the resultant zygote. We'll examine the details of meiosis 1 and 2 in a separate video.
All right. So that's big picture. So in just watching that, what's a major goal of meiosis? Reproduction. Yes. Anything else?
Genetic variation.
Yes, genetic variation, that we haven't gotten into that part of it specifically. So just think about what you learned from this video. Yes, genetic variation is a huge part. Just from what we gathered from this.
Half the chromosomes.
Thank you. So we cut the number of chromosomes in half. Why?
We'll find out. For reproduction.
Well, for reproduction. Because then when we have coming together, which does bring up genetic variation. Yes, thank you, Jesse. When you have the 2, an egg and sperm coming together and uniting, you do have genetic variation right there. But there is something else important that happens in meiosis that really increases genetic variation as well. But just the fact that you have a sperm from one individual and an egg from another individual coming together, that's cross-fertilization. So yes, that's going to be mixing DNA up there in that individual. And so if we think about that with meiosis, we've got one cell. This was something that wasn't brought up yet. One cell becoming how many?
4.
One cell becoming 4 cells. So that's important. We have one cell becoming 4 cells. We have chromosome number getting divided into 2, right? Or yeah. No. Yes. Sorry. The way I'm describing it is weird. So that's a big one right here. And then chromosomes in half. And then we also have genetic variation, which we'll get into in more detail. So these are all important reasons for why meiosis happens. If we compare that with mitosis, in mitosis, we've got one cell becoming how many? 2 cells. One cell becoming 2 cells. What about these 2 things? Do those 2 things happen? Shouldn't. These shouldn't happen. The chromosome number should stay the same. In mitosis, chromosome number stays the same. And no genetic variation. Okay? If there is genetic variation, that was a mutation that happened. So sometimes there can be when a cell divides. But that took place during S phase of interphase, when the DNA was being replicated. There might have been an error that could have occurred, and that would be a mutation. But that's not a goal here. We don't plan on this happening when mitosis occurs. And then the chromosome number stays the same. Why is it important for the chromosome number to be cut in half?
So you have the right number when the gametes combine?
Exactly. So then you're starting out with a cell that is diploid. So 2N is another way of saying diploid. The N is chromosome number. And so here we have N, and you can see it written out lowercase, uppercase. So 2N, keep that the same. And then here N, what's another way of saying that?
Haploid.
Haploid. So those are, that up here. Diploid, haploid. And so really there'd be a 1 there, right? But we don't write that in there. So, so the goal then, okay, let me add this to it next. This part's tricky because in plants, there's an extra sort of step that happens, and that brings us to alternation of generations, is that in plants, meiosis results in the formation of what are called spores. Okay? So in plants, meiosis produces spores, which then later, down the road, become multicellular organisms that produce gametes. But in animals, meiosis results specifically directly in gametes. Okay? But the ultimate goal of this is for sexual reproduction, right? So that's why we're going from 2N, from diploid to haploid, so that when 2 of these haploid cells come together for sexual reproduction, we end up with diploid again. Otherwise, if that didn't happen, can you imagine, like, how many different sets of chromosomes? There wouldn't be enough space in the cells. The nuclei would be humongous, and cells would be huge. I mean, it's crazy to think about that. So it has to be halved in order to produce these kind of cells. So but I think we did talk about this when we covered mitosis, is that there are sometimes interesting errors that can happen during the anaphase part of the process. Did we talk about that at all? All right, I think maybe we'll talk about it today. But sometimes there can be errors which can then result in different chromosome numbers than what is normal. And so we'll talk about that. Yeah.
Would that be considered like polyploidy?
Yes. that could result in polyploidy. And also with especially with plants, you can chromosome meiosis and mitosis errors can result in polyploidy and also hybridization, and even mixing of the two can result in polyploid. Polyploidism happening more readily in plants than animals, but it does happen in animals. But yeah, so we'll get into that. Alright, let's get this out of the way for now. Okay, so any questions about big picture differences between mitosis and meiosis? Any questions? All right, we'll get into details. So in talking about mitosis, we have the growth through cell reproduction, replacement of lost or damaged cells. That happens in mitosis. And we can also say reproduction, which is pretty common in plants, but asexual reproduction in plants is the result of mitosis, right? So these spider plants up here by the windows, all those little baby spiders, those little spider plants that are connected to the stolons, right? Those are the result of, those are from mitosis, not meiosis, right? They're genetically identical. So remember that. These cells here, the result would be genetically identical. So either growth of the organism or asexual reproduction. So that's just a reminder for mitosis. Now, meiosis, we've got, has to happen first in order to cut that chromosome number in half to prepare for sexual reproduction. So this part is in preparation for sexual reproduction. Okay. And so the first thing with plants, meiosis produces spores. So I'm showing the back side of a fern frond where the spores would be produced. Bless you. Whereas in animals, meiosis results directly in the production of egg and sperm, gametes. So spores are in animal, and then we've got other major groups like fungi and algae where all kinds of different things happen. It's really, this whole thing is pretty complicated. So the point is, as was mentioned, genetic variation is a big one here. And if you compare that to asexual reproduction, spider plants up here, they can do both. Spider plants can produce genetically identical little baby plants this way. They also produce flowers, and spider plants produce hermaphroditic or bisexual flowers that have both male and female parts. And the male parts are going to produce special spores that then later produce sperm. The female parts in the flowers of these spider plants will produce special spores that will later produce eggs. The egg and sperm come together, and you can have the result of a seed that then has a baby spider plant. But then that one's going to be genetically different than this parent plant. They'll probably look pretty similar. And And I have these pictures here, these two families, just getting us thinking about, well, we're humans, so sometimes I throw in pictures of humans. But just thinking about the diversity, the huge diversity you can get. And this is just focusing here on hair and skin color, eye color, and how you can have such diversity with bringing two people together and what their offspring look like. And these two pictures, I had fun pulling up because they happen to be they've had twins, and so these are twins. Obviously, they're not genetically identical twins, right? But you can see in these two examples, it just so happened that the egg and sperm that were fertilized, or the two eggs that were fertilized by the two sperm were very different genetically. Yes, question, comment. Thought I saw a hand raised. Okay. So lots of variety among offspring. So with just going back to chromosomes in general, this again showing a human karyotype, here showing human chromosomes. We can see that each one of these, well, first off, if you think about what you know from mitosis, what you learned from mitosis, when you've got the different phases. You've got prophase, the first step in actual mitosis starting up. And you've got this nuclear envelope starts to disappear. That was actually a common thing I saw in your labs, by the way, is that nuclear envelope. A lot of folks still drew that in throughout prophase, metaphase, anaphase. That sort of goes away. And then you've got those chromosomes coiling up and becoming visually apparent. And you can see them, start to see them like in prophase into later prophase. Metaphase, they line up, yeah? And then anaphase, sister chromatids separate. And if you remember from learning about mitosis, during S phase of interphase, if we just think about a single chromosome, during S phase of interphase, we have a single chromosome becoming two, but they're still connected. And so that can be a little tricky in remembering that. So we have a single chromosome that then takes on that classic chromosome look we're more familiar with. And we've got this central region here, the centromere, is kind of holding those together. So these are both, you'll hear people call both of these chromosomes, right? And but these are both chromosomes. But these are each, this chromosome here is made of two sister chromatids. Making sure you understand this will make meiosis a little bit easier to make sense of. So two sister chromatids. And so then that is actually when you can visualize the chromosomes is when you would actually do what's called a chromosome smear and do karyotyping and actually be able to count the number of chromosomes. Yes?
So then when the two sister chromatids are connected, it's still considered one chromosome?
You would still consider that a single chromosome. I know it's tricky, but then you, but really they are two chromosomes, it's just that they're connected together. What have you learned? How have you learned it?
I always learned it as they're two sister chromatids and not necessarily one chromosome, but I'm not sure, it's just like a terminology.
It's a terminology.
Thing.
It's a terminology thing. I mean, they truly are each a chromosome. but generally when you're you talk about then the sister chromatid splitting, and then you don't say the two chromosomes together splitting. So it's like it's a terminology thing, really. Anybody else want to make any comments? Yeah.
Wouldn't it just kind of be related to how we're identifying? Because it's not that a chromosome is a distinct thing. It's all chromatin. So the fact that it's duplicated, we're only referring to it as a chromosome because of the shape that it's taking during the division process. Is that?
Well, chromatin, that's a good point. Chromatin is when there's still separate chromosomes in chromatin, but they're uncoiled. So then that gets us into actually what a chromosome is. Let me go. That's a really good thing that we're going to need to talk about anyways. Let's jump ahead to that really quick here. Do we not have that in here? I might just be skipping past it. Okay, let me pull up an image. I think it'll be helpful. It was in the mitosis slides. Let me pull that up really quick. I forgot, I have another set of slides where it's mitosis and meiosis, and this is my meiosis genetics grouping. But I think this will be helpful to look at. And we, you guys remember when we looked at this? We talked about this and talk about chromosome numbers of different organisms. And we talked a bit about karyotyping. So that's what I'm trying to get to right now again. And so if you think about what a chromosome actually is a long strand of DNA that then so you have a long strand of DNA. That long strand of DNA then, when it coils up, there are histones and other components. So I'm going to say yes, you're correct, but also just to expand on that, that the chromatin is when the chromosomes are uncoiled, but a single chromosome is made up of a long strand of DNA. So you have all these separate, if you think about that, like, this could be like a strand of DNA. And so if something has 10 chromosomes, it's going to have 10 strands of DNA. And each one of those is going to coil up, and in this kind of fashion, like you see here, with the histones, which are protein molecules. So they coil up and really coil up and continue to coil up more and more and more and more and more in forming a chromosome. But that DNA then has also already replicated. So that gets complicated too. You've got a long DNA strand, and another one. They both are coiling up, but they're still connected together. And so each of those in their own right is officially its own chromosome made up of its own long DNA strand. And then during anaphase, mitosis, anaphase, they would separate out. And then once they sort of untwist, those histones, those proteins are still there, but then they open up and aren't all coiled up together anymore. DNA is still going to remain slightly coiled up in the way of so that certain genetic regions aren't just always open, ready to help produce proteins. But yeah, so it gets complicated. So a DNA strand then, or sorry, a single chromosome is made up of DNA strand plus proteins. Okay? So yeah, so having 10 chromosomes means there's 10 DNA strands. But usually when you count, this is complicated too. Usually when you're counting those DNA strands, actually let's keep that open. That's good. For and doing karyotyping, they are still hooked together as sister chromatids. So if you go back to the phases of, where are we? There we go. The phases of mitosis, and you want to find out how many chromosomes, the chromosome number of an organism is. At what phase of mitosis would you do a chromosome smear or squash and actually count the number of chromosomes? At what point?
Metaphase.
Yeah, metaphase might be a good time. Why are you saying that? Oh, who said that? I don't know. I heard from over here. Why are you saying that, Ben?
Because they're together. I don't know.
Because they're
Yeah.
together. Why wouldn't we count it here?
It's all stringed up. Like,
Yeah.
Yeah.
It's not.
Yeah.
It's not pulled up, forming those separate chromosome structures. And then why not here? Why would we do it in anaphase?
They're starting to pull apart.
Yeah, they're pulling apart. So it would be really confusing knowing like what you're counting, how many you're counting. So this is where we're getting to, Matthew, with like your number of. Would you call them? This at this point, somewhere around like end of prophase into metaphase is when the chromosome smear would actually occur, and when the counting, and it would look something like this. When the counting of chromosomes would actually happen. And so these are counting the chromosomes. But each one of these is actually made up of 2 sister chromatids. But you're still calling them individual chromosomes here at this point. So you're counting, so how many chromosomes? How many chromosomes? Seems like it would be. It seems like it would be 7
But there's
here.
8 in the middle of there. It's 8.
Oh wait, shoot!
Yeah,
I'm showing you 2 different pictures. Let's focus on A. All right. Oh no, that's
No,
fine. We'll
it's-
focus. Oh,
They're
yeah.
the same one.
They're the same. Or sorry, they're the same thing. Yes, they're both onion. I think I was just getting mixed up on how they ordered them. It was weird. For a second, I thought this was ordered in numerical order. It's not. All right, so there's. It looks like there's 8, but in actuality, there's. You'd say 16 here. But that's the diploid number. So then, if you're talking the haploid number, it would be 8. But also, what's tricky here is you're seeing that there's 2 8s. And 2 of each. Why is that?
Chromosomes are, you have a one copy from each parent of a chromosome. That's why there's like pairs.
Yeah, exactly. So if we go to here, if we have 2 haploid cells come together in fertilization, you have a fertilized egg cell, which is going to have a set of chromosomes from mom and dad, right? You're going to have a set of chromosomes from mom and dad. And those sets of chromosomes would be considered homologous chromosomes. So they're meaning what? If they're homologous, what are they? They match up, but does that mean they're going to have the same kinds of genes? Well, we haven't gotten into our genetic terminology yet, so that can be kind of tricky. They have similar kinds of genes in same location on the chromosome, but there might be what's called different alleles, different forms of that gene, or they might be the same. So if we are right here looking at a single chromosome from each parent that have the same kinds of genes at the same locations. So this is some terminology that I'm just going to throw out there now because we are talking about genetics as well. Is first off, you have a gene. A gene is a region of DNA that codes for a protein. So a region of DNA that has a recipe for a protein, for building a protein. That is what a gene is. And there's multiples of those on a long strand of DNA that then coils up. And if you imagine in this chromosome here, you've got this DNA strand is just coiled up and proteins, it's all coiled up. And you can say that this particular part of section of DNA that's all coiled up tight right there might have one of the genes responsible for eye color, let's say, okay? And we know now there's multiple genes involved with color of hair, color of skin, color of eyes. So let's say that just that region might be responsible for some sort of characteristic, okay? That is, that's a gene, and then a loci or locus is the location of that gene on a chromosome. So we can actually maybe mark that by a certain color. This region, maybe by that color, and so forth. And we can also give those genes a letter name. Like here we have the letter P for this gene, that region of DNA inside this chromosome. And you can have, so that is that the gene then would be, the symbol would be P, but there can be different forms of that same gene, which that term then is allele. So you have different forms or alternative forms of the same gene. So if we're taking a specific gene for, whether or not a pea seed is going to be yellow or green. That is a certain gene. The gene would be seed color, okay? So seed color would be the gene. Different forms of that would be yellow or green. You guys follow me on this? And then yellow would be one type of allele. Green would be another type of allele for the same gene. When talking about these things, it's kind of tricky to figure out when do you throw in different terminology as you're talking about stuff. All right, so allele then. What's an allele?
Alternate forms of the same gene.
So alternate forms of same gene. And what about locus or loci?
Location.
Locus is single. If you're talking about more than one, it's loci. So, location of gene on chromosome. Alright, so you see how sometimes this terminology can be kind of tricky in that in making sense of a homologous chromosomes, it's nice to know what genes mean, what the term gene means, what the term allele means, and loci or locus. Because homologous chromosomes are chromosomes that have the same genes at the same loci. Does that make sense? Same genes at the same regions of the chromosomes, so the same loci. What could be different between homologous chromosomes? The alleles. They might have different alleles, so different forms of the same genes. So here, and there's different ways to symbolize this. Color, letter. Here it's showing color and they're doing color and letter here. I don't know.
It's
I don't know
capitalized. That's the. I think the letter is for the allele and the color's for the gene.
but the
Isn't it?
letter is also for the gene. Letter B.
Oh, okay.
You know what I mean? They're doing
Yeah.
letter and color here.
For the gene, yeah.
So, so here we have the letter P symbolizing this particular gene. They're also using gray. And then whether the different alleles are, whether or not it's an uppercase or lowercase p. So here we have two uppercase p's, which it would be homozygous. Same. And usually the symbol for uppercase, using an uppercase letter, usually means dominant. And then here we have this particular locus is gene A, and they're both lowercase a's, telling us that those are recessive alleles. Homozygous for the recessive allele. And then here we got an example of this region between these two homologous chromosomes is B. The gene is B, also color green. And it's lowercase b and uppercase b. So dominant allele and recessive allele showing heterozygous. So this is kind of classic, though there's gonna be a lot more genes on these different chromosomes than what's shown up here, but this is just getting you to wrap around, like make sense of what homologous chromosomes are. You've got your set from mom, set from dad. Those homologous chromosome pairs will have the same genes at the same locations. It's just that some of the alleles might be different. You guys with me on that? Okay, so let's go back then. Here, you guys have homologous chromosomes down, and that's important. Let's go back to this. So here, in karyotyping, you're taking those chromosomes and we're going between a couple of these two. Actually, this is what I wanted to show. You're taking all these chromosomes, and then and what generally happens here is people will take a picture, like you have here, and then you cut them out, and then you match them up together for the homologous pairs, and like right here. And you match them up by the shape. They're different shapes or lengths, and that's how you would match them up. All right? But now, everybody look at this particular number 8. If I was to draw that out, zoom in and draw it out, it actually is like this, even though it doesn't look like it. It's just that it's so close together. Can you guys see that? It's so close together that you can't tell it's actually made up of 2 sister chromatids. But it is. So you have a single chromosome duplicated during S phase of interphase, and then during metaphase, prophase of metaphase, you can actually see those chromosomes. That's when they're pulled out, counted, and that's when they're actually 2 sister chromatids stuck together. All right, so how many chromosomes do humans have? 46.
Mommy says I have 47.
What's that?
I was telling myself, my bad.
No, some people might. Some people might have 47. Some people might have 45.
Yeah.
Yes, that does happen. Sometimes you could have somebody who just has an X chromosome, and sometimes you can have 2 X's and a Y. Never are you going to have somebody with just a Y, because the X chromosome has important genes on it for life, for being able to survive and grow and live. The Y chromosome doesn't. Not saying the Y chromosome is not important. It is important.
You can live without a Y, right?
Yeah, that's what I'm saying. You can. Otherwise, everybody in here with just 2 X's would not be alive right now, right? You don't need to have a Y. And you don't need to have 2 X's. You can have a single X chromosome. Yes?
Doesn't the X have something to do with mitochondria?
As far as, yeah, that sounds familiar. But it's other stuff too, which I don't have memorized all the different important things that the X chromosome provides. But it's a lot. It's a lot. And hence also why X chromosome is quite a bit larger than the Y chromosome as well. It's got a lot more important genes on it. All right, so, oh, and you can see it right here, that size difference. There's that cute little Y. All right, so, where was I going with this, you guys? Jeez Louise. All right, so we've got homologous pair. And notice, each of these chromosomes is really made up of 2 sister chromatids. That's why we don't say, when we're looking at this, we don't say there are 46, or no, we do say there are 46 chromosomes. We don't say, wait, what's 46? My brain's 92. We don't say there are 92 chromosomes here. But actually there are, if you think about the sister chromatids. Hopefully I'm not trying to confuse you. I'm just trying to get you to think about that, how complex these chromosomes are here. And you can say here there's 22 homologous pairs. Why is it just 22 and not 23?
Because the X and Y are homologous.
Yeah, the X and Y actually are not homologous. They're not, because to be a homologous chromosome pair, to be homologous chromosomes, you have to have the same what? Genes in the same loci. And they don't. They're different chromosomes. All right. So, in meiosis, big picture, this is what that video clip we start out with just talks about pretty much. Shows that you have chromosome, we're just following one homologous pair through this process. We have a replication of the DNA, or DNA synthesis occurs during the synthesis part of interphase, the S phase of interphase. And so each of those is going to duplicate. We have, during meiosis 1, we have pairs coming together. And this part the video didn't talk about, which really increases the genetic variation that results in meiosis. There's something really cool that happens right here, and that's crossing over. So during crossing over, we have sections of DNA cross. So if I'm going to erase this part. You guys can help me out here. Here is our cell. Oh, actually. We're gonna run out of space. Here's our nucleus. And here's all of our chromatin. Here's the nucleolus. What phase of the cell cycle is this cell in? It's an interphase. Then we jump right into, if we're just thinking back to the phases of mitosis. What's the first phase of mitosis?
Prophase.
Prophase. Interphase is not the first phase of mitosis, that's the cell cycle. So let's jump ahead to that first phase of, now we're going to switch our brains here to meiosis. But luckily the terminology is the same. So we're going to jump into prophase. Meiosis, there's two major parts of meiosis, meiosis 1 and meiosis 2. Meiosis 1, we're just going to remind ourselves we're in meiosis 1 if we say prophase 1. Okay? So if we say prophase 1 here. It's really just the same as prophase and mitosis here. What do you think's going on? The nuclear membrane goes away. We can kind of go like this. It's kind of going away here. It's gone. And we have all that chromatin then to coil up. Bless you. We have starting to look kind of wormy. That chromatin is, we have the DNA coiling up with those proteins. We have actual chromosomes developing here. All right, so we've got chromosomes. And what happens is we have homologous pairs binding each other, coming together. So let's do a few. I'm going to just do 2 homologous pairs here. Okay, here's 2 homologous pairs. And they pair up. Before, the homologs are just all over the place. But if meiosis is going to be happening, they come together and pair up. Not necessarily at the middle specifically, but they will. So, but what's happening here? They pair up, and they are paired very closely together. And if we give let's add some color here to the chromosomes. Red and green. All right, so this is showing there's the different color here is symbolizing different alleles. All right, so red and green are the same gene, just different forms of the same gene, so different alleles. And I'm going to do it over here, this one. All right, is everybody with me here? These, during prophase, we have a crossing over that occurs. And so that's not really obvious until we have what's called anaphase, which you guys know, when the pairs actually come apart. So let's move here to metaphase. Then we're going to draw this out again. Now they're lining up in the middle. So in metaphase is really similar to what you saw in prophase, except now they're lining up at the middle. Whoops. Red. And I'm going to show this one over here really close. And these two over here are very close to each other. And they're sort of overlapping. And at this point, crossing over has actually happened, but it might not be super obvious for us at this point because they're so close together. But I'm going to make it obvious. So crossing over means sections of DNA have actually swapped. So crossing over sort of happens right in this area. Prophase 1, metaphase 1, those pairs are together, sections have swapped. And so I'm just showing you 2 homologous pairs, 4 chromosomes, but think about our cells and how many we have, and some plants have 70 or over 100 different chromosomes. So in plants, this could be happening like at a much higher, and also it could be happening in different parts as well. Multiple crossing over events can happen with the same chromosomes. All right, so crossing over right around here. And then you've got anaphase 1. Now, in mitosis, what happens in anaphase?
The sister chromosomes separate.
Yeah, so let me show you guys those spindle microtubules. Sister chromatids separate. In anaphase 1, pairs separate. So, if pairs are separating, we've got all these colors again. Then you've got this one over here. Just making sure. And this one's going to be what color? Green. And the other green is going to go where? Way over here. And then red. And so forth. All right, so this is meiosis 1. Instead of drawing every single step out for meiosis 2, the result of meiosis 2 is it goes through these phases again, but we're going to add a 2 at the end of each one. And meiosis 2 is actually the same as mitosis at that point in the way of sister chromatids separate. So if we fast forward here, fast forward to fast forward, jumping past telophase, prophase, metaphase. We fast forward through these steps, then jump into anaphase 2. What would we have? Yep. But the important thing is, now all of a sudden, we've got I'm running out of space. I made myself a little big. Okay, so we're kind of skipping ahead here for the sake of time and drawing all these out. For this one, this is going to split in telophase, and we're going to have the result over here. Maybe we should do metaphase first, huh? Pretend this is metaphase, and then I'll reset. Alright, now we gotta do the colors again. This part takes a little while. So we've got it's okay if they kind of swap a little here. So that's metaphase 2. So let's talk about here the difference between metaphase 1 and metaphase 2. Here we've got pairs lining up in metaphase 1, and then sister chromatids, right? Individual single file lining up in metaphase 2. Then if we skip ahead to anaphase, then those that are sister chromatids are going to be pulled apart. And so, can you guys then tell me what we end up with? So these are going to split down the middle there. If we have telophase and cytokinesis. So we're going to end up with what are we going to have over here? Haploid cells. And this one, if we go along with this one right here, what's going to be here? The green and the orange. We're following following this through and I didn't get mixed up. Did I do that right? Oh, I see what I did wrong. It's something weird here. They should all be different. These two, I should reverse these two, maybe. Sorry guys, those two were kind of mixed up. Alright, so then we should have I still did it weird, didn't I? Was it correct before?
Looks correct, man.
No. I think it was actually right originally. Sorry, I was actually correct originally. All right, and then this one. And hey, bless you. All right, sorry that takes a little while to work through it all. I'm sorry I cut some of it out, but in the end, every single one of these cells then is haploid or diploid.
Haploid.
So we've got they're all haploid. Because someone is allergic to meiosis. So these are all haploid. And what I started doing is drawing a couple of them the same. They should all be different. So none of them are the same. And when you compare them to the original, which the original pre-duplication. The original pre-duplication would be I may have had it and erased it, our original one over here. Would look something like this. All right, so this is what the original looked like. We have one diploid cell. In the end, we have 4 haploid cells that each look very different from the original parent cell and each look very different from each other. In animal cells, if these would be gametes. They'd either be egg cells or they would be sperm cells. But in a plant, they are all spores. That then, as we get into alternation generations, those spores then become plants that then produce egg and sperm. So when we covered mosses, everything we looked at in that Bryophyte workshop, those were all haploid. That whole moss structure or liverwort structure you looked at, those were haploid, multicellular haploid structures that developed from a spore that then are going to produce egg and sperm by mitosis. That is sort of tricky to get our head wrapped around that. So here we have a wake robin plant that showing anaphase 2 and 2 different cells. So it's sort of tricky in that I don't like to generally show telophase 1 happening specifically or cytokinesis because often that doesn't happen necessarily. You don't have 2 complete cells that then divide into 4. Generally, it's all kind of happening in a single kind of cellular structure like we see here. That then in these 2, this would be the result of anaphase 1, and then anaphase 2 in each one. And then telophase 2, then cytokinesis would then happen, producing then separate actual pollen grains, which are spores, okay? So here we can see the chromosomes that are starting to then uncurl, and you can actually sort of make out the DNA in each of them. And they're starting to form new cell walls between all of them. So you may hear 2 homologous pairs together are actually considered a tetrad, because it's in reality 4 sister chromatids that are each 4 chromosomes then. Crossing over occurring results in what are called recombinant chromosomes. So recombinant, you'll hear recombinant DNA technology, where you're taking DNA from one thing, putting it into something else. Something happens within our own cells with crossing over. You have DNA swapping, whole DNA sections. All right, so what's the benefit of crossing over?
Genetic variation.
Yeah, it just really increases that genetic variation. You're going to have genetic variation already with sexual reproduction because you have the mixing of 2 genomes. A genome from mom, a genome from dad, getting mixed. But no 2 egg cells or sperm cells or plant spores are going to be the same. And that's because of crossing over going on. This is sort of this is actually a real image of crossing over happening in grasshopper chromosomes. So you can see it happening in multiple places. So in the lecture online, these slides I think will be helpful in just showing what we learned from cytokinesis in plant cells, where you actually have the cell wall pulled into it. And it depends on the cells. This is actually producing the production of spores. And of course, not gametes. So finally, before we go to our break, I want to quickly go over alternation of generations, which I mentioned we kind of already covered in that Bradfite workshop, and we'll cover again multiple times. You'll get tired of hearing it, or you'll get very comfortable with it, hopefully, later in the semester. Yes?
I know it's been a while since we went over genes, but did we ever go over recessive genes? I don't remember.
It hasn't been that long. It was just like really fun. And we haven't really gotten into genetics too much, but recessive versus dominant would be different alleles. So we did kind of talk about it, just that, but we didn't specify what happens with dominant versus recessive, but we will. So, can you wait? Can we go through that later? We have to go through it now?
Yeah.
Okay, well, I'm gonna say,
Okay.
sorry, we're gonna move
got
on.
vetoed.
All right, so just wrap your mind around what's happening here with alternation of generations, okay? If you think about pine tree, okay, let's think about a pine tree. Pine trees are what are called sporophytes. And the sporophyte, P-H-Y-T-E, sporophyte actually means spore-producing plant. So that big pine tree that you see is a sporophyte. And it has the gametophytes living in its cones. So they're much tinier. In flowering plants, the gametophytes live inside the flowers or inside the pollen. So they're much smaller. And the sporophyte in a flowering plant is the flowering plant. Is the sunflower plant, is the cabbage plant, or whatnot. So that's a sporophyte. If we're talking about bryophytes, like mosses, like we already learned about, that main structure we're familiar with seeing is actually considered the gametophyte. If a sporophyte is a spore-producing plant, what's a gametophyte?
A
Gamete-producing plant, yes. So here, this is a really simple way to just make sense of what's happening here between gametophytes, sporophytes, what's haploid, what's diploid, where meiosis is happening, where mitosis is happening, okay? That's what I want you to get down now. So, and this is a pretty easy way to think about it. Like we got it on the board, and I don't like that marker. This is better. So, when I have 2N, what does that mean? Diploid. So if we think everything under this line then is diploid, and we have the sporophyte. The sporophyte is diploid. And I mentioned in pine trees, the sporophyte is the pine tree. That is diploid. And it's in then the cones where the gametophyte is actually produced. Okay? So if we have a pine cone right here, it would be inside of there, would be where gametophytes are produced. Okay? And gametophytes are haploid. If we're talking moss, here's our little moss down here. And actually, I'm going to put it up here. Here's our moss. The moss is a gametophyte. And then here's our pine. I did what? The moss here. When the moss produces these cool structures you guys learned about, remember those? Those are actually the sporophyte of a moss. The gametophyte of a moss is that fuzzy carpet structure that we're familiar with. This would be inside a pine cone is where you have the gametophytes for pines. So it's very different. So as we get into the major plant groups, you're gonna get you're gonna have to do a flip-flop with your brain in thinking about this. All right, so gametophytes are, and what this shows is just showing that sporophytes and gametophytes are both multicellular. That's the whole point of how this kind of diagram is showing you that. But it's the sporophyte that is going to undergo, by way of meiosis, to produce what? Meiosis in plants produces spores. And the cell that undergoes meiosis is obviously within the pine or within the moss, or within the sporophyte of the moss, is diploid. And it's going to undergo meiosis to produce 4 haploid spores. Because the sporophyte is the spore-producing plant, it's going to produce spores by way of meiosis. Now, those spores might land in a suitable location. If they do, they undergo mitosis. Those spores will undergo mitosis and form a multicellular gametophyte. Is that gametophyte, if haploid spores undergo mitosis to produce a multicellular structure, is that multicellular structure haploid or diploid?
Haploid.
It's going to be haploid because mitosis results in identical cells as the parent. So mitosis occurs, we have a multicellular gametophyte, which may be a small structure inside of a pine cone, or it becomes a larger structure like this moss or liverwort. And so then there's multicellular component within each of those that then produces gametes. The gametophyte produces gametes, and it produces the gametes by way of mitosis. So we have then gametes, egg, sperm, and we need to make sure that's still under the line that differentiates diploid and haploid. And then those gametes get together and form a diploid zygote. So that is, in a big picture, what's happening with alternate generations. If humans did this, we would be considered the sporophyte. We would produce spores inside us somehow. And maybe we'd cough them out. That'd be weird, right? We're like coughing out these spores. There's like, I'm coughing and you guys are seeing this like, and then like spore cloud come out.
Fertilization happens on a table somewhere.
Yeah, well, it needs to probably land in something
Imagine getting
nice
fertilized because some idiot
moist,
coughed in your class.
nutrient rich. So these spores land somewhere suitable.
A dish sponge.
A dish sponge. Over here in the sink. It lands in the sink. And then starts to multiply, divide. The spore is haploid, produced by meiosis. My sporophyte body does not produce gametes. It does not produce egg and sperm. It just produces spores that go off and land somewhere. Or with really advanced plants, like a gymnosperm or an angiosperm, maybe I'm producing female spores that stay inside my flower or cone. Okay? But less advanced, evolutionarily advanced, I'm not saying advanced like better, but moving along evolutionarily, the spores then just all get released. And they land somewhere, become multicellular organism that then produces egg and sperm by mitosis or meiosis? Mitosis. So, gametes are produced by mitosis, not meiosis in plants. So if humans did that, our little multicellular gametophyte, well, it wouldn't be phyte because it's not plants, it'd be like gametozoa, or gametozoa, I like that. Those organisms, multicellular organisms that are haploid, they're the ones that get to produce the egg and sperm and be involved in that. We just get to cough up the spores as the diploids. That's crazy to think about it like that. But that is exactly what happens in the plant world. And it happens like that in some other major groups as well.
Don't jellyfish do something similar except they don't switch between haploid and diploid?
They do.
They
Jellyfish
have
do some really interesting things.
The like floating form, and then they have the one that it like kind of just grows on the seafloor.
Like, so you're between the polyps and the medusa.
Yeah.
The medusa,
Because the medusa is the
it's like the
sexual
swimming.
is the sexual form, and then it makes the polyps, and the polyps they make more of the asexual reproduction.
They
So
do
is it
some
similar, but-
really interesting things.
But as far as I know, they don't switch between haploid and diploid in the different generations.
I don't think they do. They don't do that. They still produce. I don't think they actually produce multicellular haploid structures that I'm aware of, but I could be wrong. I don't think any. I don't think anything. I don't think
But they
anything
do
in the
have
animal group.
alternate generations between asexual and sexual. Because the polyps don't do sexual reproduction, it's only the medusas.
I keep thinking about sea anemones. Because sea anemones do this interesting thing where they're floating around for a while until they get a suitable location. Then they do everything in their polyp form that I'm aware of. But jellyfish will do this thing where they can revert back to an immature state. And maybe is that where you're going with that?
No, I can look up the
Going back and forth. But yeah, I don't, it's in the animal kingdom, we don't have a going back and forth between the diploid and haploid.
Yeah.
That doesn't happen, that I'm aware of. But that's really interesting. All right, so questions about alternative generations? Yes?
So, like with a pine tree, it will make this other, like, haploid kind of thing. That's the pine cone, and then the pine
Inside the pine cone.
Inside the pine. How does the pine cone get the sperm and the egg to, like, go together? Is there like a male and a female pine cone, or?
Very good, exactly. That's it.
There
That's what
is.
happens.
Okay.
There's female cones that actually in these scales, the cone scales, the female cones have a special place inside of them where they have a special spore that becomes the female gametophyte, and it stays inside. Whereas the male cones are smaller, and they have these little scales that will produce, they're called micro spores. Inside a microspore, you have division taking place. Well, you have the mother cell microspore, and that one divides. Then you have the microspores, and then they become pollen grains.
Okay.
So
and those make their way
yeah,
to
so then the
female-
pollen, the sperm cell travels inside of the pollen grain.
Okay.
And so some of that, the microspores, they then become pollen. And then those pollen grains by wind will land inside of here, and the pollen grain kind of what germinates, and then the sperm inside of that pollen grain then connect with the egg that's inside of here. But the egg was developed by a female gametophyte. So the male gametophyte, there's a male and female gametophyte. It's complicated. It's really nice to start out with a moss life cycle because it's much more simple, and you're not dealing with microspores and megaspores and male gametophytes and female gametophytes. You're just dealing with that nice green carpet moss that we're familiar with. There's some parts that produce sperm at the top, some parts that produce eggs at the top, and the sperm will get splashed into where the egg is, and out of that, the sporophyte develops.
Oh.
And produces spores for the next one. But it's okay. It's honestly, if you get it, that is amazing. So because it is really good. And I know we're just kind of scratching the surface of it. So if you don't fully get it, that's okay, because we'll be covering it again. So we'll end, and then we'll go on a break after this quick. Did you guys watch the life cycle video?
What were these? The question is,
I'm going to skip ahead to a little bit of a different one. Just hopefully help make this a little more concrete.
This projector supposed to be on?
bryophytes include the various mosses and liveworts that are found commonly growing in moist, shaded areas in the hills. Bryophytes are also called amphibians of the plant kingdom because these plants can live on soil but are dependent on water for sexual reproduction. The plant body of bryophytes is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by unicellular or multicellular stage bears the sex organs.
Alright, so this is the communiphyte or sporophyte. This is the green stuff we're familiar with saying. So the gametophyte in mosses is the dominant generation of the alternation generation of mosses, okay? In a pine tree, it's the pine tree. That's the sporophyte, right? It's a sporophyte. Alright, so it's different. Let's see what happens here. Everybody focus in here. Are these haploid or diploid? Haploid.
The sex organs are multicellular. The male sex organ is called antheridium. They produce the flagellate antherozoids.
Or sperm. Alright, so some of the terminology here is funny, but the visuals are good. So the top part of the gametophyte, you have special sexual structures produced. The male ones are going to produce the sperm by mitosis or meiosis. They're already haploid. So mitosis. Sperm are haploid. The gametophyte plant is also haploid. And on top of some areas, there's female sexual structures produced that are actually called antheridia, or archegonia, sorry. The male ones are antheridia, the female ones are archegonia. They're going to produce the eggs. And the eggs stay inside, kind of like a womb sort of structure, whereas the sperm you'll see get released.
A female sex organ called archegonium is flask-shaped and produces a single egg. The antherozoids are released into the water where they come in contact with archegonium. The antherozoid fuses with the egg to produce the zygote. Zygotes do not undergo reduction division immediately. They produce a multicellular body called sporophyte. The sporophyte is not free-living but is attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division or meiosis to produce haploid spores. These spores germinate to produce gametophyte.
So what was kind of jumped past
But is
here,
attached to the photosynthetic.
Okay, so this is the gametophyte down here. This is the sporophyte. What's the ploidy level of the sporophyte? Very good. Haploid. Very good. Diploid. I need to wake up here. I didn't get enough sleep last night. All right. So gametophyte, haploid, N. Sporophyte, diploid, 2N. Yes? So the sporophyte's going to produce the spores by mitosis or meiosis? Meiosis. Very good. I'm glad you guys are more awake.
Synthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division or meiosis to produce haploid spores. These spores germinate to produce gametophyte.
So
Let's solve some important questions for medical exams. Question 1.
No, we won't do that part. So I just wanted to show you those visuals. All right, let's go ahead and meet back here in 20 minutes. Ready to